So first of all, RNA transcription is occurring in what part of the cell? And this you may want to type onto your web page. RNA transcription occurs in the nucleus. So this process is occurring in the nucleus of the cell. Now, when we did our comparison of DNA and RNA, we talked about the differences between DNA and RNA. We said that DNA was double-stranded, RNA is single-stranded. We talked about if DNA has deoxyribose sugar, RNA has ribose sugar. So those are some of the key differences. Basically, there were different types of RNA. We talked about messenger RNA, which is what we'll focus on right now. Later, we'll talk about tRNA, and there is a third type called ribosomal RNA. So today's focus will be on mRNA, and if you'd like to type on your website or write down that mRNA means messenger RNA. So it's taking a message from the nucleus to the ribosome. So mRNA means messenger. So messenger RNA is going to go through these steps to get the information coded from the DNA and take that to the ribosome. In the ribosome, the next process we learn about is protein synthesis. So if I get the information from the DNA, I can actually make the proteins in the ribosome. So why can't DNA just take its information, leave the nucleus, go to the ribosome so they can build the proteins? Why? Why would it die? You're on the right track. We talked about how it couldn't leave. Correct. If you have one set of blueprints and that blueprint is allowed to leave the secure area and there's an accident and something changes or a letter gets miscoded or something gets moved, you have now changed that entire blueprint for that person, for that cell, for that organism. So we want to keep DNA in a safe place. So if we're thinking about a cell, if this is a cell, if this is the nucleus, we want messenger RNA to go into the nucleus, copy the information, and then it's going to take it out to the ribosome. And that's where the protein is going to be made. But it's a copy that we're taking. So you could think about RNA transcription as if we are the ribosome and we're the people that can build the things that we need. Or you could think of the ribosome as the shop, right? A place where they could assemble information. What part of the school would we say would be like the nucleus? Right, so the office would have important information. In the office, what would be similar to a chromosome or to chromosomes? Teachers leave the office? So we could say the office secretaries are like the chromosomes. They have all of the information for the building. It could be a file cabinet where the papers are kept in. So let's work with the file cabinet. You could use the people there, but technically they could leave. Files, let's say they have to stay in the filing cabinet and they can only be looked at in the office. So if I went into the filing cabinet, if you think of the filing cabinets over here, if I was to pick a specific chromosome, I'd have to pick, say, the black filing cabinet. To pick a specific gene, I would have to pick one of the drawers, take the information out of there. It's then like I'm opening up that file folder and I'm seeing the information. If that information is typed, how would I have to write it down so I'd be allowed to leave? Right, so if it's typed, could I just make a photocopy of it and leave? Because that would be too much like the original. What would you suggest I do? Okay, I can make a Prezi. A Google presentation, I could write it down. As long as the information is coded slightly differently, still the same order, still the same info, but the format is a little different, then I'm allowed to leave the office because I'm not taking an original blueprint. Then I can find my way to the ribosome, which would be the shop or the classroom. I would walk in here, say, here's the information, and the rest of us would build whatever that file or set of instructions said. So that's kind of like a quick comparison to what this process is about. So step one, we're inside the nucleus. Messenger RNA is going to the nucleus, and it's searching for a specific gene. The one thing that I would add in right here is before it looks for a specific gene, I would say it looks for a specific chromosome. So if you'd like to write directly on this page, if it hasn't been updated in the duotang, it's okay if you do that. So messenger RNA goes into the nucleus. So if I was a messenger RNA, I come into the room. If every grouping of tables is a chromosome, I find the right chromosome. And then here I'm going to look for the right gene, which might be one of the people that's here. Question? Okay. What you may want to jot down there is, if it makes sense when I say, if I was the messenger RNA and I'm looking for table sets and tables represent chromosomes, and I pick one chromosome, then I look for a specific gene. If you need that analogy, take a moment to put that in. The only other thing it adds here is that a gene is a section of a DNA molecule. So it's only a portion of that chromosome. Gene would contain things, an example would be the gene for eye color, the gene for hair color, the gene for straight, curly, or wavy hair. Okay, so the gene controls a specific type of trait. Now, the picture that I have located here, this part here and here is the DNA strand. So the messenger RNA is coming in, and it's taking a look here at this strand, and it's deciding where it should go. It finds that specific gene, so it finds this portion right here, and it decides in step 2 that it has to unzip that portion. What do you think the DNA is still held together by? Right, so there's still hydrogen bonds in here. So there's a hydrogen bond in between. We have split it apart in this section here. What do you think breaks those hydrogen bonds? Right, so an enzyme comes in at the specific gene. The enzyme goes through, breaks those hydrogen bonds apart, exposes just that portion of the gene. So we have this split apart. Are we okay to step 2? Okay. Now, this is when the messenger RNA actually goes into the DNA molecule. And we need to remember how DNA pairs up. We said A always goes with T, C always goes with G. In RNA, however, adenine is pairing with uracil, so uracil is what makes RNA different in the code. C still goes with G. So if along this outer edge here, you have the letter G for DNA, it pairs with a C in messenger RNA. If you have a T in DNA, it still pairs with adenine and RNA. If you have an A in DNA, that's where it pairs with a U in uracil. So basically, wherever RNA would have had a T, a U is substituted in. Are we okay with that? In your notes, have you made a connection between C and G still always together, but adenine is going to pair up with uracil? Just in RNA. If it's DNA, and that's a good question, because if it's DNA, it's still going to pair up with a T, and that's how the nuclear membrane would know that that's not allowed to leave. So messenger RNA is going to be the only thing that uses that U. C and G together, a T in DNA with an A in RNA. So it goes in, finds a specific chromosome, finds a specific gene, breaks the weak hydrogen bonds using an enzyme. Messenger RNA goes in and pairs up. And it says step 3 here, once part of the DNA molecule is exposed, one strand of the DNA is used as a template. So it picks one side. Does it technically matter which side of the DNA molecule it picks? Yesterday when you formed, new people joined in, did it make a difference in the code? No, because you're still pairing up in the same order. So it doesn't matter which side they pick. So in this diagram, the capital letters represent DNA. The lowercase letters represent messenger RNA. And in here, And you'll notice that AUG is at the beginning. And when we get into protein synthesis, we'll talk about nitrogen base pairs or nitrogen bases in threes. A group of three is called a codon. Every gene starts with AUG because it codes for the amino acid methionine, which is the starting point. So messenger RNA nucleotides come in, temporarily match, and temporarily is a key word there, match with one side of the DNA, which is used as a template. And we can spot the differences because there's a U, so that's how we know it's RNA. Once that full length of the exposed DNA has been copied, the newly synthesized or newly made RNA pulls free, so it's kind of like a little train. When you get all of the cars in order, then that train can pull out, leave the nucleus, and where does it go? Right, so it goes on its way to the ribosome. DNA after that point goes back together. That means transcription is complete, and the RNA is now at the ribosome ready to start the next process. So the whole purpose of RNA transcription is to get the information from the DNA to the ribosome so it can actually make things happen in the cell. So it can create proteins that allow you to do the things you need to do to survive. So let's do one more time, just check the key points. So if you've been typing the key points in, we'll do a quick review of that. Then we are going to pick a hand signal for each of those steps so that you can begin to associate what this refers to, what this refers to. And so if you get to an exam, you can run through those hand signals in your head or act them out, and that will give you the steps that you need. So step one, where is it happening? Right? So the first thing you should have written down or noted if you're adding something there would be it happens in the nucleus. Messenger RNA comes in and it searches for a specific chromosome and then a specific gene. So come into the room, I look around for the right table set. At that table, I look for the right person who has the information that I need. Once it finds the right gene, it breaks the hydrogen bonds between the nitrogen bases using an enzyme. So an enzyme breaks those hydrogen bonds. The only part that splits apart is that gene. So a specific section is split apart by an enzyme. The next step, and we will go through these multiple times in the next few minutes, is one side of the DNA is used as a template, and the letters for messenger RNA come in and pair up. So make sure that you have A in DNA now pairs with a U in RNA. C and G are still the same. So we have the new way that it pairs up. Once it has the right code, it leaves and goes to the what part of the cell does messenger RNA want to take that information to? Correct. So it goes to the ribosome. The DNA, which is still split apart or bubbled at that section, what happens to it now? Goes back together. It is not changed. So we haven't damaged anything. It's not changed. It is ready for the next time information needs to be copied. Transcription is complete. Questions? What do we think the purpose is? Not quite to make the protein. Making protein is the protein synthesis step. It's getting the information from the DNA to the ribosome so it can make the protein.